topic, the stringent response, and this is the usual disclaimer that it is meant only for students of life sciences and it should not be used for any other purpose, copied or posted on any site without my permission. And some of the pictures in the presentation have been taken from public's domain website or some popular books to make this topic easy to understand, and the credit for such pictures remain with the original publisher. Okay, so what we have learned is that prokaryotes or lower organisms, they have rather limited resources. Very often they live oblique survive in hostile environment, hostile in terms of environmental condition as well as nutrient supply. And they deal with scarcity or fluctuating nutrient supplies. This is very important. Scarcity is there one and fluctuating because they don't know what is going to be available next moment. So they have to deal with that. And how do they do it? They have learned to live under these conditions. They have learned to survive under these conditions because they have learned to live, or they have learned optimal utilization of their available resources. Optimal utilization of their available resources. This is important. Okay. And what do? They exercise economic prudence. Okay. Flexible approach. So we have discussed these in last 3-4 lectures, that they show massive flexibility, huge flexibility from metabolizing one metabolite at this moment to metabolizing other the next moment. So they are very flexible in that, and they do so very swiftly. They mount a very rapid response to a given situation. So because of these three things, economic prudence, flexible approach, and swift response, because of these three, they can survive under these difficult situations. But sometimes the nutrient availability is, particularly that of proteins, becomes very poor. Okay. So in that circumstances, in spite of exercising these three, economic prudence, flexible approach, and swift response, they have to shut down some of their activities, a range of activities rather, as a last resort to survive. And this particular phenomenon is called a stringent response or stress response. I'm repeating it once again. Sometimes the nutrient availability, particularly of proteins, becomes very, very poor. And in that particular situation, the bacterium as a last resort shuts down a range of activities to survive as a last resort. This is called a stringent response. Okay, so what happens during stringent response? In stringent response, the rate of protein degradation increases. Okay, why protein degradation? Because it is the protein, it is the amino acids which is really scarce. So for synthesizing new protein, it requires amino acids. So amino acids are not available from the medium, from exogenous sources. So what does it do? Its own non-essential proteins are being degraded so that amino acids required for synthesizing essential proteins can be made available. Second thing is synthesis of stress proteins and proteins which are required for amino acid synthesis, there increases. Synthesis of these two things, proteins which are required for synthesis of amino acid, enzymes which are required for synthesis of amino acids, their synthesis increases, and stress proteins, their synthesis increases. Stress proteins, oxidative and osmotic stress survival genes, they are upregulated. Those genes which are involved in managing the oxidative stress or osmotic stress, those genes, they are upregulated, means their expression increases. So this is the thing. So all these activities, rate of protein degradation increases, synthesis of proteins which are required in amino acid biosynthesis, and synthesis of proteins which are required in management of stress. Those synthesis increases, and Genes which are involved in oxidative stress and osmotic stress management, those genes are upregulated. And at the same time, a few things are downregulated. What are the things which are downregulated? Synthesis of rRNA, ribosomal RNA, and tRNA. It decreases, and it decreases significantly. There is almost a tenfold drop in the synthesis of ribosomal RNA or transfer RNA. Similarly, messenger RNA synthesis, it also decreases by as much as threefold. So there is a massive drop in the synthesis of synthesis of RNAs. Ribosomal RNA, tRNA by tenfold, and mRNA by threefold. Similarly, biosynthesis of carbohydrates, nucleotides, and lipids, it also decreases. So as a last resort, bacteria is involved. Bacteria does these things. It upregulates a few things and it downregulates a few things. Okay, what is the trigger? What basically triggers this stringent response, or what we are calling it as a stress response? The trigger is an uncharged tRNA at the A site of the ribosome. So now you are aware what is the A site. Right? Typically at the A site you have transfer RNAs which are charged with amino acid. You call them as amino acid tRNAs. But an uncharged tRNA, because amino acids are not there, okay, if an uncharged tRNA occupies the A site, that becomes the trigger. What we have seen, as I just told you, generally a charged tRNA, amino acid tRNA, occupies the A site during protein synthesis, and the protein synthesis continues. But due to shortage of protein nutrients and particularly amino acids, there are not enough amino acids available in the pool. Okay, and as a result of that, a uncharged tRNA, occupies the A site. Shown over here. This is an uncharged tRNA. This is an uncharged tRNA which is occupying the A site. There is a codon here. So codon is for an XYZ amino acid. So what you would have expected, a tRNA carrying amino acid XYZ occupying the A site, but that's not the case because amino acids are not there. So an uncharged tRNA comes and occupies the A site, and this is the trigger. So you see on the P side you have peptidyl tRNA, a tRNA carrying the already synthesized polypeptide chain, and you have a deacylated tRNA at the E side. This is a usual picture in protein biosynthesis, but as soon as an uncharged tRNA occupies the A side, this becomes the trigger. Now it is also known that antibiotic stress, when there is a lot of antibiotic in the medium, that antibiotic stress is also known to trigger the same response. Same response is elicited when there is too much of antibiotic in the system. So when you have this situation, what happens? When you have an uncharged tRNA or there is an antibiotic stress, okay, what has been found out is that it leads to accumulation of two unusual metabolites. Bacteria accumulates these two unusual metabolites. And what are those two unusual metabolites? One is guanosine tetraphosphate. Guanosine tetraphosphate, also designated as PPGPP. And the other one is guanosine pentaphosphate. Guanosine pentaphosphate, also designated as PPPGPP. These two metabolites, these two unusual metabolites, they are found to be accumulated in bacteria during stringent response. Together, these two metabolites are also designated as P in bracket, then PP, GPP. Okay. So instead of writing both these two, you can simply write this just one, P in bracket followed by P followed by P, GPP. It indicates that you are talking about guanosine tetraphosphate or guanosine pentaphosphate, both, right? These are also called as alarmones. These two metabolites are also named as alarmones. You must remember this word, alarmones. Okay. And how are they synthesized? They are synthesized by the action of these two genes. There is one gene called REL-O, and recently they have found out There is another gene called SPOT. These two gene products means the protein they code for. Usually they are enzymes. Okay. These alarmones are synthesized by the action of these two gene products or the proteins or the enzymes synthesized by coded by these two genes, REL-O primarily, and recently there is one more that has been found out is called SPOT. The proteins they are synthesized, they are coded by these two genes are called as stringent factors. They are called as stringent factors. As I just said, REL-A gene product is an enzyme. It is an enzyme. It is also called a stringent factor, but it is an enzyme. And the name of the enzyme is guanosine pentaphosphate synthetase. Guanosine pentaphosphate synthetase. Okay, the name of the enzyme or stringent factor is PPPGPP or guanosine pentaphosphate synthetase. What does it do? It catalyzes transfer of a pyrophosphate group, transfer of a PP, pyrophosphate group from ATP to 3-hydroxyl of GTP or GDP. The recipient molecule can be GTP, recipient of pyrophosphate group can be GTP, or recipient of pyrophosphate group can be GDP. Okay, and where is this pyrophosphate comes and attaches? This pyrophosphate comes and attaches at 3-hydroxyl group of the GTP or GDP, and it synthesizes. If it gets attached to GTP, it synthesizes a pentaphosphate, and if it gets attached to a GDP molecule, then it synthesizes a tetraphosphate. Okay, let us see that reaction also. Okay, so this is the reaction that I have tried to show you over here. So you have an ATP, which is the donor of pyrophosphate, and if it donates that pyrophosphate to GTP molecule, the reaction is catalyzed by guanosine pentaphosphate synthetase. So what do you do? What do you get? It results in the formation of a guanosine pentaphosphate molecule. If the donor molecule ATP donates a pyrophosphate to a GDP molecule, okay, then the resulting molecule will be a guanosine tetraphosphate. So if the receiving molecule, recipient molecule is GTP, you get pentaphosphate, and if it is GDP, you get a tetraphosphate. Okay, and both the reactions are catalyzed by stringent factor REL-A gene product, which is nothing but which is a guanosine pentaphosphate synthetase enzyme. You can also get guanosine tetraphosphate from guanosine pentaphosphate. This is the way. A specific phosphate catalyzes the reaction. What does it do? It removes one phosphate, and it is aided by ribosomal proteins, particularly this elongation factor EFTU and EFG. You know, this is an elongation factor, and this is a translocation factor. And some other ribosomal proteins are also involved along with the enzyme phosphatase. So guanosine pentaphosphate can also be converted to guanosine tetraphosphate. So there are two ways guanosine tetraphosphate can be synthesized. One is by this reaction, and the second is by converting pentaphosphate to tetraphosphate in presence of enzyme phosphatase and ribosomal proteins, particularly EFTU and EFG. Okay, what is written in some textbook is that this is the major route, that guanosine tetraphosphate is synthesized from guanosine pentaphosphate. Okay. Okay, so what is the sequence now? The sequence is that REL-A is activated. Okay, REL-A gene remains dormant. It remains switched off. Okay, it is not getting expressed. It gets activated, it gets expressed, it gets switched on only when an uncharged tRNA enters the A site. Okay, there is a provision that what will happen, what bacterium will do when there is an acute shortage of proteins in particular. So when an uncharged tRNA enters the A site, this has been considered as a trigger. REL-A gene gets activated. Okay, ribosome, as I just showed you in one of the earlier slides, ribosome idles, means stops at the A site on the mRNA, waiting for a charged tRNA. Ribosome idles on the messenger RNA, waiting for what? Waiting for a new amino acid tRNA to come and occupy the A site so that protein synthesis can continue. But since amino acids are not there, there is a protein scarcity. Ribosome idles means ribosome pauses over there. So presence of a uncharged tRNA causes a change in conformation of the A site. If A site is occupied by an uncharged tRNA, its conformation changes. And this conformation change in the site, that apparent A site, is transferred to L11 protein of LSU. Now, we have seen when we studied the structure of ribosomes that large subunit, it is made up of both RNA as well as 31 different types of polypeptides. And one of the polypeptides which makes large subunit is called as L11. So that conformational change in the A site is transferred to L11 polypeptide of the LSU, which in turn causes activation of REL-A. That conformation change in L11 protein serves as a trigger, and the REL-A gene expression gets switched on. REL-A gene gets activated. So REL-A and guanosine tetraphosphate, they function to dislodge the uncharged tRNA from the A site. Both of them together, they're responsible for putting that uncharged tRNA, which has entered the A site, outside the ribosome. So they dislodge it from the A site, and that uncharged tRNA comes out of the ribosome. So how this expression of REL-A gene, causes tenfold drop in rRNA synthesis, tRNA synthesis, threefold drop in messenger RNA synthesis. A probable mechanism has been, what you can say, worked out. What was worked out is, what was seen is that guanosine tetraphosphate binding to RNA pol1, RNA polymerase. Binding of guanosine tetraphosphate, which is that unusual metabolite which gets accumulated during stringent response, it binds to our RNA polymerase, and prevents the complex from this complex formation, prevents opening of the DNA double helix. Okay. When we are studying transcription, what we found out is, what we studied was that RNA polymerase binds to DNA double helix, and it melts, it unwinds about 17 base pair of DNA. And this transformation from a closed promoter complex to an open complex is a key event in transcription. Okay. So this is how RNA polymerase will come and bind to the promoter. Okay. How does it identify promoter? It identifies promoter with the help of those -10, -35 consensus sequences, and it unmelts, it unwinds about 17 base pair of DNA. This is what is that 17 base pair of DNA. Okay. And this transition from a closed complex to an open complex is the key event in transcription. So basically, this guanosine tetraphosphate, it interferes with this. Guanosine tetraphosphate, it interferes with this. I have shown you here. It interferes with this. It doesn't allow that complex. It doesn't allow opening of this helix. PPGPP means guanosine tetraphosphate binds to the site where RNA polymerase binds to this site on DNA where RNA polymerase binds and forms a complex. Okay. So what does it mean? It means that guanosine tetraphosphate also binds to the promoter. Promoter is the site where RNA polymerase binds. So guanosine tetraphosphate also binds over there where RNA polymerase comes and bind. And this binding of guanosine tetraphosphate prevents the open complex formation. It prevents conversion of a closed complex to open complex. And as a result of that, transcription cannot take place. And that is why you see such a massive drop in mRNA synthesis, tRNA synthesis, and ribosomal RNA synthesis. Moreover, this particular event means slowing down of conversion of open complex, closed complex to open complex, it also slows down elongation step of transcription by forcing increased pausing of RNA polymerase. Okay, so as far as this is concerned, this prevents initiation. This is the initiation event. So it doesn't allow initiation of transcription. As a result of that, ribosomal RNA, tRNA, and messenger RNA synthesis stops. But those transcription processes, those transcription events which are already underway, even their speed, gets substantially reduced, because this particular event, binding of conosine tetraphosphate at the promoter, somehow prevents, somehow forces RNA polymerase. It takes higher number of pauses. There are increased pauses of RNA polymerase, and as a result of that, even those transcription events which are underway, they also get slows down. So cumulatively, the effect, what do you see? You see the transcription means rRNA, tRNA, and messenger RNA synthesis reducing considerably, significantly. As a result of that, all those enzymes which are required for biosynthesis of carbohydrates, lipids, nucleic acid, those enzymes become less, and as a result of that, the synthesis of these biomolecules, decreases, and that is what bacteria wants. Because bacteria is, has adopted stringent response as a last measure to survive during protein scarcity, particularly the amino acid availability becomes really scarce. So this is what is called a stringent response. So that's all in this stringent response.